Hello and welcome to my academy. In the previous lectures, we discussed about an overview of the photosynthesis, chlorophyll, and other accessory pigments. We also discussed about the light-dependent reactions of the photosynthesis. Now, we are here to discuss the light-independent reactions of the photosynthesis, or Calvin cycle. In plants, carbon dioxide enters the interior of the leaf through pores called stomata and diffuses in the stroma of the chloroplast, the site of Calvin cycle reactions, where sugar is synthesized. These reactions are also called light independent reactions because they are not directly driven by the light, rather uses the products of light dependent reactions. Before we proceed, let's have a quick look on light-dependent reactions of photosynthesis. The light-dependent reactions convert light energy into the chemical energy. The goal of the light-dependent reactions of the photosynthesis is to collect energy from the sun and break down water molecules to produce ATP and NADPH. These two energy-storing molecules are then used in the light-independent reactions. Now we are going to discuss light-independent reactions or Calvin cycle. Calvin cycle, also known as reactive pentose phosphate cycle or C3 cycle. The Calvin cycle was elucidated by Melvin Calvin and Andy Benson. For the discovery of this cycle, both were awarded with a Nobel Prize in 1961. The light-independent or Calvin cycle can be discussed under the following three headings. Number one, carboxylation or the fixation of carbon dioxide. Number two is the reduction phase. And number third is the regeneration phase. Now we will discuss all the phases of the Calvin cycle one by one. In the carbon fixation phase, carbon dioxide is incorporated into a 5-carbon compound, ribulose 1,5-bisphosphate. The enzyme which catalyzes this step is ribulose 1,5-bisphosphate carboxylase, or Rubisco. The product of the reaction is a 6-carbon intermediate which immediately splits to form two molecules of 3-phosphoglycerate, also termed as glycerate 3-phosphate. Thus, for every 3 molecules of carbon dioxide that enter the cycle, 6 molecules of glyceraldehyde 3-phosphate are formed. This was all about the carbon fixation or step first of the Calvin cycle, in which we get 6 molecules of 3-phosphoglycerate. The second phase of the Calvin cycle, that is reduction phase. The phase involves the reduction of 3-phosphoglycerate into glyceraldehyde 3-phosphate. First, the 3-phosphoglycerate is phosphorylated at the expense of one ATP molecule to form 1,3-bisphosphoglycerate. This step is catalyzed by a phosphoglycerate kinase. The later molecule is then reduced by glyceraldehyde 3-phosphate dehydrogenase to form one molecule of glyceraldehyde 3-phosphate. Thus, 6 molecules of ATP are utilized in phosphorylating 6 molecules of 3-phosphoglycerate to 1,3-bisphosphoglycerate, and 6 molecules of NADPH are consumed in reducing 6 molecules of 1,3-bisphosphoglycerate to glyceraldehyde 3-phosphate. Thus, from the reduction phase of the Calvin cycle, what we get is the 6 molecules of glyceraldehyde 3-phosphate. The last phase of the Calvin cycle, the regeneration phase. In the regeneration phase, a molecule of the glyceraldehyde 3-phosphate may leave the Calvin cycle to form the triose phosphate, which later on synthesizes the carbohydrates. This triose phosphate contains 3 carbon atoms. The remaining 5 molecules of the glyceraldehyde 3-phosphate contains only the 15 carbon atoms, and it is this glyceraldehyde 3-phosphate which is used to regenerate ribulose 1,5-bisphosphate. Regeneration of ribulose 1,5-bisphosphate is an active process. For regeneration of ribulose 1,5-bisphosphate, 1 ATP is required. After that, 3 molecules of carbon dioxide is again incorporated into a 5-carbon compound, ribulose 1,5-bisphosphate. In this way, the cycle will run again. Again, the triose phosphate is formed. This triose phosphate will combine with the previously formed triose phosphate to form the 6 carbon compound glucose molecule. If you like the video, then do subscribe and share. Thank you.